Hello and welcome to this video on bioprospecting. This is the process of finding active compounds in plants, animals, and nature that can then be used in a pharmaceutical capacity. It involves screening massive numbers of relatively random samples and finding a possible fitting purpose. These are then commercialized via patents. Bioprospecting is one reason to consider environmental conservation. The diversity of life means many possible novel products. These samples can come from the wild, traditional medicine, and expanding the current pool from predictive models. These can be genes, enzymes, proteins, inorganic molecules, and more from special plants, animals, fungi, bacteria, and other organisms. There are many possible targets and products to explore. This field of study is mired in some controversies. The three major sources of novel biocompound development are wild or randomly occurring products. An example of this is amphibians, who often produce surface toxins or proteins. Some of these have been shown to be promising antibiotics. Traditional medicine is another area. This involves plants and inorganic products used traditionally for illnesses or health benefits. Matcha tea is one example. It is used in energy drinks. More obscure or specialized applications such as pharmaceuticals are also developed this way. Unfortunately, this is also sometimes called biopiracy, and that will be described later. But for now, understand that it has caused quite a few problems. Finally, there are projects that are working to use the large databases of active compounds with known uses and using computer power to predict possible similar performing compounds. This can then screen a massive database of unknown or products with no known use for a match or similar structure. These are the three major sources of novel bioactive compounds. As has been described previously, you cannot patent a thing that is already known. This is why bioprospecting exists. The companies or institutions doing this are not looking to find a new product that they can patent. Instead, they are looking for an application or process that can be patented. Occasionally, a refined, specially isolated, or synthesized version of the product can be patented, but this is an exception to the rule. A well-known but less well-recognized example is morphine and opium. The opium poppy is like tea. It was used for centuries, if not millennia, before it became a common medicine. It was used extensively in Asia Minor. It was also found in Neolithic European caves. It was commonly used leading up to around 500 BCE, after which it seems to have dwindled in popularity. During the Islamic Empire, it gained prominence once again. This led to the Crusades, which brought opium back to Europe. This was eventually taken to China, and the Opium Wars occurred. The pure morphine product was isolated in 1804 by Friedrich Wilhelm Adam Sertiner. This created a known dose, an important aspect of good pharmaceutical science. It was the first isolated product of its kind. Through the 20th century, opium was used on an as-is basis. This meant that one batch of extract from the opium poppy could vary drastically from another. In the 20th century, it was superseded by synthetic and semi-synthetic versions that were more pure, more effective, and generally cheaper. Screening projects are the most fundamental approach to bioprospecting. These are also the least efficient approaches. This involves taking plants that could be anything. More often than not, they are samples from regions with to a large body of either new or poorly described species. These have their contents extracted with a variety of solvents. Common examples are alcohols, nitriles, and water. This creates a range of active compounds that fall into a narrow range of electrochemical properties. This helps to figure out the how and why of its function. This is also when the structure is established. This is done with chemistry and biological methods. The products are then investigated in cell and animal models. Generally cells first to see if it is extremely toxic, to get an idea of what sort of dose should be used, and if it has a specific function, such as being a good anti-cancer agent. This extraction approach produces a variety of compounds that have different electrochemical properties, which will alter how they are delivered to the body. This extracts a variety of compounds that have different electrochemical properties. This will in turn dictate how they can be delivered. For example, something may not be soluble in water, and therefore must be put in a character molecule that will allow it to be injected or taken orally. Something may need to have a very slow release, and therefore must be consumed orally. And other factors that over time will dictate just what sort of medication is used and how it is made. Traditional medicine is the next approach. This relies on traditional uses that often lack substantive medical evidence. These are your traditional supplements like cactus juice. An investigator will use this information as a launching point for their investigation of the medical properties claimed by the traditional use. Database mining is the final way to find novel products that can be commercialized. There are institutions and companies that retain huge records of molecule structures, functions, and synthesis pathways. This includes records of products that have been made but have not found a purpose. Think of it as banking future products. The way this works is sort of complicated, and so the simplified version will be given instead. It starts with a program that can pull each molecule out of the database, or can match one against what's already in there. This can be examined for its properties like shape, size, and charge. This can be fed into an algorithm that creates a probability of it then acting or matching a similar desired product's properties. It acts almost like a search engine for your library of desired product features. It works because modern technology allows the storage of both complex information on many features and the ability to interrogate it in a timely manner. Considering the example of morphine, more modern examples might be easier to understand. An example of this includes willow bark and aspirin. It was synthesized and manufactured in 1899. Before that, salicylic acid, which is the active compound of aspirin, had been used medicinally for decades, centuries, and again, possibly millennia. It was made from the willow tree, another plant that were rich in this particular compound, and there is evidence that the ancient Egyptians may have been using it. In 1853, Charles Frederick Gerhardt, a chemist, treated acetyl chloride with sodium salicylicate, which produced acetyl salicylicate. Later, in the 1800s, other chemists found a more efficient way to produce this compound. In 1897, researchers for Bayer were investigating acetyl salicylic acid to find a less irritating option. Eventually, they found a way to do this, and by 1899, they had created what is now known as aspirin, and were selling it. Through a variety of decisions over the years, the name itself has lost much of its original protected meaning. This course of development deviates from the original product significantly. Not only that, but it alters the traditional use, more so in the late 20th century. Going into the 20th century, aspirin was losing a lot of popularity as a painkiller, but in the late 20th century it had a revival, particularly as a preventative treatment for heart attack and stroke, an application unknown in the early history of its use. A more current example is Taxol. From 1967 to 1993, almost all Taxol produced was derived from the bark of the Pacific yew. The Pacific yew had one major drawback that prevented its use as a chemotherapy agent. It grew in a limited environment, took years to mature, and ultimately was too scarce and yielded too little. The compound that is Taxol was identified through a pure screening program run by the US government. Not only did they identify the original product, but then a species related to the Pacific yew that could be processed to achieve a semi-synthesized version of the same active ingredient. These three examples are generally without the contention some other examples have. This is where the earlier mentioned biopiracy needs explanation. Biopiracy is, in simple terms, when a company or researcher uses information or knowledge from traditional sources or cultural practices to find a novel use or product that an indigenous or regional group has used for extended periods. They then monetize it for commercial purposes. There are two examples of this. The first supports a case for native claim to some of the intellectual property recognition, and this is the Maya ICBG Bioprospecting controversy, which occurred between 1999 and the year 2000. The ICBG group is the International Cooperative Biodiversity Group, and they were accused of being engaged in unethical forms of bioprospecting, particularly NGOs and indigenous organizations. The fundamentals of the argument are that the ICBG claimed to be documenting the ethnobotanical knowledge of the indigenous peoples, and this allowed them to figure out whether or not there was a possibility of developing this same body of knowledge into medical products, particularly plants used by the indigenous groups. But the work they've done is based upon traditional knowledge that they haven't fully recognized in terms of who is responsible for the original work. The second is a case for the opposite. The rosy periwinkle dates from the 1950s. This is a case about a plant native to Madagascar, but it had been introduced into many other tropical countries. As a result of it being so widespread through so many tropical countries, different cultures had developed beliefs about its medical properties. This gave researchers a place to start, where they could obtain local knowledge from one country and plant samples from that country, or from another. The use of the plant for diabetes was the original thought here. Researchers were under the impression from traditional use that it could be used in the treatment of diabetes. However, what they found was that it was effective in the treatment of Hodgkin's disease and leukemia. The Hodgkin's lymphoma chemotherapeutic agent vinblastine is derived from the rosy periwinkle. This is a very different product to the diabetes medication that they were chasing, and this is an example of where traditional medicine hasn't led to the final product. There are certain laws to protect the environment and developing countries from these biopiracy raids. The leading convention is that called the Convention of Biological Diversity that came into effect in 1993. The major foundation for this is that it secures rights to control access to genetic resources for countries where those resources are based. This sort of thing would have protected the Mayan people from the ICBG controversy. It does this by better balancing the ability of a less developed country to leverage their resources and traditional knowledge when it comes to bioprospecting. It has created a set of requirements that each international patent is bound by. This is then reinforced by trade law. It sets down a series of rules between researchers and countries about the benefit sharing and, in many cases, can lead to royalties for those developing countries. And then you have an example from India. This is called the Traditional Knowledge Database. Due to previous cases of biopiracy and to prevent more in the future, the government of India converted traditional Indian medicine from the ancient manuscripts and other similar inaccessible formats to an electronic version. And this resulted in the Traditional Knowledge Digital Library in 2001. This makes it a lot harder for a company to patent a discovery if there is already evidence of its common use in this database. Bioprospecting is an important avenue of new drugs in the past and going forward. It is likely to provide the next generation of novel antibiotics. There are some concerns about the weaker economies and less developed countries being taken advantage of, but overall, there is work towards a reasonably equitable arrangement for this form of investigation. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.